Welcome, students of higher secondary second year, who those who have biology as one of your subject. And today, from the Department of Zoology, as a part of the subject biology, I am Dr. Chakravarty, and I would be today talking with you about the principles of inheritance and variation. If you go through your book, you would find that it is in the 7th unit and the 5th chapter. So, this is about the various facts and laws of inheritance and the variations in genetics. But then there are various parts to it. I would today discuss about the Mendels Law of inheritance, as you can see in the first slide. Now, I think you are all aware that it was Gregor Johann Mendel, an Austrian monk actually he was, but he was very much interested in science and he wanted to study medicine. So he went to Vienna, Austria, and but could not finish his course. Later he became a monk, but he did not leave his urge to experiment with natural things, and he took to his passion the work of finding out the answers, or rather, he was trying to find out the answers as to why things happen and how do they happen. So he selected the sweet pea, the pea plant, and he found that some of the plants were growing tall, some were dwarf, some had yellow color spot, some were having green color spot. So there were variations, there were character differences in the pea plant and their components. So it was intriguing to Mendel that why such thing happens. And you would understand with time that that was the first seed, the question that came to his mind that led to today's genetics, or as we know it in modern science. Well, Gregor Mendel, or Gregor Johann Mendel, he actually saw the pea plant, studied them, and selected 7 traits, 7 characters. And he was lucky in a way that the characters he chose were in different pairs of chromosomes. Well, we shall discuss that with time, but in your first year, you have studied about the chromosomes, and you are well aware of the fact that chromosomes are in pairs, homologous chromosomes, that is. So, he was lucky that none of the 7 characters that he chose were in the same homologous pair, that no 2 of them or 3 of them were in the same pair of homologous chromosome. So as such, it became easier for him to understand and to do the experiments that he intended to do. However, before going into details about the Mendelian laws, we need to refresh some of our ideas, and we need to look into some of the terminologies that we are going to use while we discuss genetics per se, and specifically the Mendel's laws. Now, one of such terminology is inheritance. The very word inheritance is something which is transmitted, which is derived, or which we receive from our ancestors. Here, in biological terms, inheritance refer to the characters that the offsprings, that the children acquire from their parents. And what characters? Specifically, these are the genetic characters, these are the biological characters, and of course the physiological characters, because all the subsidiary characters are commanded by the genetic traits. Now, when we talk of Mendelian inheritance, which is otherwise known as Mendelism, is dependent or is based on the tenet, on the principle that each gene is meant for one character. Of course, in our later knowledge, we shall see that how modern understanding of the genetics has now given away to a much complicated and much wider understanding of character, genes, and their transmission. However, from Mendel's time and what is now known as classical genetics, we have to take it for the sake of learning that each expressed character is dependent on a pair of gene which is present in the chromosome. Now, although it was Mendel who did all those experiments and he very meticulously noted down all that he found and all those results that he actually deciphered. But it was very interesting to note that his works from 1865 to 1866 did not lead him to a conclusion. He was rather not very conclusive in his statement that what really did his experiments were showing. And it was later in the 1900 that scientists like Correns, De Vries, Henry Sutton, and others, they redid Mendel's experiments. They went all through the acts that Mendel did and wrote down in his texts and notes. And with time, these experimenters found that yes, what Mendel wanted to show through his experiments were indeed correct to the results that he predicted. But then the question still remained, what are the inferences? So, my dear students, let us now again try to understand some of the terminologies, because unless we understand the terminologies, we won't be able to appreciate the proper understanding of the laws which post-Mendelian scientists came up to after re-evaluating Mendel's work, rather redoing Mendel's work and deciphering the same data. Now, for that matter, what Mendel called as traits are, today we know, we call them as genes. Now, a question might arise in your mind, what is gene? Or where is the gene? In your HS first year classes, you have understood the structure of chromosome and the structure of DNA. But in the structure of DNA, which you find is a sequence of polypeptide chain, or in another way we can say that it is a polypeptide base pair chain. The moment I say base pair chain, the word pair is important because we have learned that two nucleotides on either side joined together by hydrogen bonds, be it 2 hydrogen bond or 3 hydrogen bond as the case may be, in case of adenine and guanine and cytosine and thymine being the nitrogenous bases. There are billions of such sequences linked by phosphate or phosphodiester bonds. Now, the human genome is in weight 3 picogram and in length it is 93 centimeter. Now, yes, you have heard it right, it is 93 centimeter. Now, a 93 centimeter, 11 nanometer double strand of thread is within the cell, not only the cell, it is within the nucleus of the cell. And the cell is not visible to our eyes. I am talking about a normal body cell, not about an ova or a avian ova. So, in those microscopic cells, even more microscopic nucleus, you have a double stranded structure which is by weight 3 picogram, by length 93 centimeter, and by width 11 nanometer. Now, that is what we have called it the chromatin fiber. And on chemical analysis of the chromatin fiber, we have learned that it is actually formed of base pairs of nucleotides. And we know by now that nucleotides are structures made up of pentose sugar, phosphate, and nitrogenous bases, the adenines and the guanines, the cytosines and the thymines, that is, the purines and the pyrimidines. And of course, you know about the attachment sequence, that A is connected to T and C is connected to G in order to keep the width of the DNA constant and all of those things. However, if we are aware that a DNA is formed of nucleotide bases, then again the question arises, where is the gene? There is nothing written on the chromatin thread or on the nucleotide bases which tell us this is a gene. Yes, there is nothing written, but there is a chemical signature. That means the portion of the chromatin fiber or certain number of nucleotide bases which work together expressing a protein is a gene. Any section of the DNA which is capable of expressing itself in terms of transcription followed by translation and protein synthesis, you term that particular section a gene. A gene again is not of equal length. Theoretically, you may say, then that means one base pair can express itself and it can be a gene. Well, that would be the smallest unit in the sequence of billions of units. But for a gene which expresses, for a section which expresses, when you call it a gene, it could have 100 base pairs, it could have 1000 base pairs, it could have lakhs of base pairs. And the number of base pairs in different genes are different and varies widely. But the Human Genome Project, which is complete, has found out that the entire human genetic constituent have about 25,000 genes. And there are more inert non-expressing areas than the expressing areas. And we know now that we call the expressing areas as the exons and the non-expressing areas as the introns. So the genes, because don't you forget ever that the chromosome or the chromatin fiber are in pairs, and we call them homologous pairs. So every gene has its fixed position in a certain chromosome at a certain locus. If you consider two pencils side by side, and each of the pencil has one marking at the center of the pencil, then both this marking on both the similar pencils would be alleles of each other. That means these are partner genes of the same locus working on the same function. As for example, the gene for human body color, dark brown, yellow, white, is located in the 13th gene. In the 13th chromosome, and it has a certain locus. The gene for gender determination, the TDS gene, is located in the Y chromosome. Likewise, every character has a gene and it has a certain locus. Locus meaning a certain area, a portion, a designated chair for the gene to be in the designated chromosome. Now, of these two partner genes which express the same character, one might be dominant and the other might be recessive or both may be equal in strength. Now, when we talk of dominant and recessive, let me give an example. If we talk about the character length, we are not talking about 4 feet or 6 feet, we are talking about length. Length is the character, and in a person, there would be two genes of length, one from the father and the one from the mother, acquired by that person, and they would be interacting amongst themselves. One of the gene could be dominating the other. The gene which would be dominating the other allele, the other partner gene, for the same character length would be expressed and is known as the dominant gene. And the other gene or the other allelic gene for the same character length, which will not be expressed in that particular person, will be known as the recessive gene. But that does not mean that the recessive gene has been lost. No, it will remain there. So, I give an example here. You have Amitabh Bachchan who is long and Jaya Bhadur who is short. But you have seen that both Abhishek Bachchan and his sister are tall. That means the gene for greater length has been dominant over the gene for shorter length, and the children of Amitabh Bachchan and Jaya Bhadur, here Jaya Bachchan if you may like it, are tall. That brings us to the second term, genotype and phenotype. Now, phenotype is what you see, that what is been expressed. Abhishek Bachchan is tall, but internally he has one short gene and one long gene. That means his genotype is heterozygous, not homozygous, not of the same quality. Had it been tall from both sides, we would have called Abhishek Bachchan a homozygous tall individual having two tall genes, and therefore he is a pure tall. But he is not. He is a heterozygous tall. He is not a homozygous tall. So the phenotype is the physical appearance, and genotype is the actuality that is within the genic characters of that individual. Now, I have already discussed with you about dominant and recessive. And you know by now that among all the characters that are expressed phenotypically, now you might not be careful about this and think that phenotype means what you see. No, even the enzyme that we secrete, the growth of our cell, everything is a phenotype. Characters which are functional are also phenotypes. Not only your looks, your hair, your nose, your height, or your skin color. These are visually seen. But there are many more characters which your body is expressing, which are internal. But all of these are phenotypes. And in all of these phenotypes, expressions, there is one gene which is dominant and the other which is recessive. Because we are here discussing only dominance and recessive. However, I would like to give a subsidiary understanding at this point that not that always two genes which are alleles, one is dominant and the other is recessive. It is not like that. It may so happen that of the two competing genes or the two alleles, both are equally dominant. Then what will happen? Will they fight? Yes, they fight in a certain way, and we call it the codominant situation. And what is the expression? You might have seen a cow with red and black color. Some part is red, some part is black. That means in those areas of the body where the black gene got dominant, it became black, and in those part of the body where the red gene became dominant, the color became red. So we call such a situation a codominant case. Again, we see something where a red flower is bred with a white flower, and the offspring that we get is a pink flower, neither red nor white. Color is a character. One gene has red expressive character and the other has white expressive character, but the offspring is having an intermediary pink color. Why it is so? The answer lies that both of these genes are incompletely dominant. We have learned about dominance, we have learned about recessive, we have learned about codominance, but in this case, both of these genes have tried to express themselves. However, they have failed to express themselves completely, and one could dilute the other partially, and therefore none could retain their individual characters. This is what you have, an incomplete dominance, and the expression is in between the two. All right, so we have, in case of a dominant expression, you will always have either two dominant genes. Here it is given by capital B, because we are considering the capital B is dominant over small b, or it could be capital B small b, because as I said, capital B is dominant over small b. Only when it is small b, both are recessive, double recessive, then only the recessive character will be expressed. Well, genotype and phenotype, I think we have understood this. And also we have understood what is heterozygous and homozygous. Having similar alleles for genes is homozygous, and having differential genes in their potency or expression is their phenotype. Is the genotype, and the phenotype is actually the expression that these genes create in the individual's body. Well, after understanding these fundamental terminologies of classical genetics, let me tell you, there were 7 characters that Mendel had picked up from the sweet pea, and these 7 characters, as you can see them, one was flower color, which was purple or white color, the flower position, either axial or terminal, whether the flower is a terminal flower or is an axillary flower, the stem length was there, a tall plant or a dwarf plant, seed shape, round or wrinkled, seed color, yellow or green, pod shape, flattened or constricted, and pod color, green or yellow. So these are the 7 characters, contrasting characters that Mendel took. However, we would see that his characters were luckily in 7 different pairs of chromosome, thereby avoiding what we now call as intergenetic interaction. All Mendel's experiment were on intragenic interaction, which is classical genetics, and not intergenic interaction, often called epistatic interaction. Now, that brings us to the 3 important experiments and the subsequent development of what we today know as the Mendels Law of Inheritance. So, once Mendel was done with his experiments, but he could not come to any conclusion, it was later Boveri, Sutton, and McCarthy, and all of these people, they had actually gone through his experiments, and after rediscovering what he did, in honor of Mendel, postulated 3 important laws. The first law is the law of dominance. What Mendel did, he selected tall pea plants and dwarf pea plants and kept on self-breeding them, and found that after some time the tall plants keep on producing tall offsprings while the dwarf plants keep on producing dwarf offsprings. After some time, he thought that the tall should be a pure tall and the dwarf should be a pure dwarf. He set up an experiment where a pure tall was bred with a pure dwarf plant. Here you can see that the pea plants that he used were having, we know that TT genes, capital T, and small t. The capital T genes, when would segregate, would become one capital T in one sperm, another capital T in another sperm, and the small t would become one small t in one sperm and another small t in another sperm if the dwarf is a male. But if the male is tall and the female is dwarf, for our convenience, then we would have a situation where there will be two capital T genes in the sperm and two small t eggs. Here B has been used for your understanding. And we see that when the two individuals are bred, all the offspring that are resulted are tall. Why? Because the capital T is dominant over small t. Because you have got two capital T's and two small t's. When one small t links with another capital T and the other small t goes with another capital T, always there is a capital T which is dominating the small t. So in the first filial generation, that is the first offspring line, you have children or offspring which are tall phenotypically, but genotypically they are heterozygous with one capital T and one small t, or you may take it a capital B and a small b, doesn't matter. So the result is, from this we can say that when two individuals differing in one pair of contrasting character, I'll repeat it, when two individuals differing in one pair of contrasting character are crossed, then the dominant character is expressed in the F1 generation, and that is known as the law of dominance. Here you can see, in a hybrid union, the allele which expresses itself phenotypically is the dominant allele, while the other allele which fails to express itself phenotypically is the recessive. Well, that brings us to the first law. That means the dominant allele always expresses. Now we can focus on to the second law, or the law of segregation. Now if you focus your thought onto this fact that in the first experiment, one pure tall was bred with one pure dwarf, and the children, that means the parents, were pure tall and pure dwarf. But the F1 generation, the first filial generation, they were tall nonetheless, but heterozygous in their genetic makeup. Genotypically, they were not pure. However, they were all tall. Now, if we breed two of these heterozygous offsprings and create the third generation, the F2 generation, that means we would be breeding a capital T small t individual with a capital T small t individual. The result would be very interesting, that all of those individuals who were phenotypically tall were actually hiding a dwarf character in them, and as the individuals were producing gametes, these contrasting characters segregated from each other, separated from each other. And the result is we see that a 3:1 tall to dwarf offsprings are formed. That means we get one pure tall, two heterozygous tall, and one pure dwarf. Now, where did the dwarf come from? Because the parents here were both tall. Because the parents were tall phenotypically, but genotypically they were impure. So the gene for dwarfism, or the recessive gene, got a chance to segregate out and combine with a dwarf gene to produce that one individual which is like its grandmother. Just a way of saying so. Like the parental generation, one of the parents was dwarf. So from that experiment, the law that was postulated says that when two individuals, focus on the law, when two individuals heterozygous genotypically but similar phenotypically for a particular character are crossed, then the recessive characters segregate from the dominant character and breed true, producing a double recessive individual. So that is what the law of segregation, or the second law of Mendel, states. Here you can see the first law and the second law. In this chart, you can see that the parental generation, one parent is pure tall and the other parent is pure dwarf. So the F1 individuals, which are from the first generation, filial generation, they were all tall. However, you can see that it is a capital T and a small t in the F1. They are tall, but they are not pure tall. But when we breed two of such capital T small t F1 individuals, we get the F2s, where you have got one pure tall. You can see the first left corner in the 4 square box, which is capital T, capital T. Then you have a capital T small t followed by another capital T small t, and there is only one small t small t. In the bottom box, you would see that the ratio is 3 tall is to 1 short. This 3:1 is the phenotypic ratio for monohybrid cross, and it justifies our first law of inheritance and the second law of inheritance. Now we can come to the third law, or the law of independent assortment. The third law, or the law of independent assortment, is the most interesting of all the laws, because it is the root of diversity, it is the root of variety, and because of it the world is so diverse, so varied, and so much full of different forms of organisms. Now, thus far, we have understood the crosses with one character, but no individual are of one character. There are thousands of characters, physical characters, physiological characters, mental characters. There are so many more. Biological characters are varied, and each character has its own genic root. And what is happening is these characters have a peculiar nature of mixing independently and making independent choices, of going in pairs of their choice and expressing what they want to express. So let us first see the law as such. The law says when two individuals differing in two or more pair of contrasting character. You can see here that we are differing from the earlier statement when we said when two individuals differing in one pair of contrasting character. Here we are saying that when two individuals differing in two or more, now this more can be many more, as many more that can be. When two individuals differing in two or more pair of contrasting characters are crossed, then the individual characters assort independent of each other amongst themselves. This means they don't remain attached to the partners they were earlier in the parental stock. They go away from their earlier partners and make new partners and express themselves in the F2 generation as new traits or new characters. The new character here is not something which is novel to the organism. It happens at a later time, which is mutation, but gradual. But at every generation with birth, there will be some variation in the nose, some variation in the color, some variation in the skin. Everything will be changed, but the skin will be there, the nose will be there, the ear will be there. But you would find something is changed, something is like the other one, something is changed, and it matches somebody else. So those are the changes that keep on occurring. When many of such changes accumulate, we term them as mutations, and these mutations leads to variation. Variation leads to isolation, and isolation leads to speciation. Thereby, new species formation in this world had actually occurred this way. So now let us see that to do this experiment, we would go back to Mendelian experiment, and what he did. He selected two pea plants. One was having round and yellow colored seed. The seeds were round, full and round, and they were yellow. So two characters, round seed, yellow colored seed. The other plant he selected was having green colored seed but wrinkled. The seeds were not full, they were wrinkled. Okay. Now I'll come to this. You can see that he selected round yellow and green wrinkled seed. Now the gametes that formed from the male and the female side are tabulated here in the Punnet's box in the horizontal and the vertical axis at the top and at the left margin. So the round yellow and the green wrinkle are the parental characters. They were the parents. But when the offspring of those parents were formed, which parents? Round yellow and green wrinkle parents. All the offsprings were round yellow, because round was dominant over green and round was dominant over wrinkled and yellow was dominant over green. Now all the F1s were round yellow, but as in the case of monohybrid cross, we had seen that all the offsprings were tall but the dwarf gene was hidden. So also in this case, when round yellow parents were crossed with green wrinkle parent, all the children of the offsprings were round yellow, proving that round and yellow were dominant characters to green and wrinkled. But when two such F1s, that means second generation individuals, were crossed, a wonderful thing happened. 16 offsprings from the Punnett square, 16 offsprings are tabulated, and we see that 9 of the individuals formed are parental type, round yellow. Another one individual is parental type, which is green wrinkled. And you can see the round yellow in the upper and the left margin and shown by yellow round structures. And the last right hand lower corner is that of the other parental type, which is green wrinkled. Now what about the other 6? 3 are green round and 3 are yellow wrinkled. Now you might wonder, none of the parents were green round or yellow wrinkled. They were not green round or yellow wrinkled. They were green wrinkled and yellow round. Then how did these two characters come into existence? This is because the green gene, the yellow gene, the round gene, and the wrinkled gene, although they were together in their F1 generation, when they came to the F2 generation, they started assorting themselves independently and making new friends. So yellow made friendship with wrinkled, round make friendship with green, and 6 out of 14 offsprings were new types. So that is what is the proof of independent assortment. To understand that, we have to look into the explanation that it can be explained with the help of a dihybrid cross involving the characters of cotyledon, as I said, yellow round and round wrinkled shape, and of course the color, okay, that is yellow and green. Here the genotype of both these things are being given by capital Y and small y for yellow, and capital R and small r for wrinkled. So what we see, the formation, and here you have in the first line the capital Y, capital R is the parent, one of the parent, and the other parent is small y, small r. That means the one parent is yellow round and the other parent is green wrinkled. In the first two lines you can see the genotypes, and when they were crossed, all the F1s hybrid resulting from the fusion of these gametes would be heterozygous yellow round, because capital Y dominant over small y, capital R dominant over small r. But then when these F1s, which are capital Y, capital R, small r, two of these are crossed, as you can see in this chart, we have the result which I have just discussed with you. This result, which is 9 yellow round parental, 1 green wrinkled again parental, and 2 types green round and yellow wrinkle, 3 and 3 are non-parental. This 9:3:3:1 is what is known as the phenotypic dihybrid ratio. 9:3:3:1 is the phenotypic dihybrid ratio for classical genetics. Of course, the genotypic ratio of this particular cross of this dihybrid ratio would be 1:2:2:4:1:2:1:2:1, which is 12-24-12-1. That means you have to remember this word 12-24-12-1. That means you write first 12, then you write 24, then you write 12 and ek, and put the is to sign in between. So that will be 1:2:2:4:1:2:1:2:1. Total is 16. And this genotypic ratio talks about the type of genotype, because if you count how many capital R, capital Y are there, you will find only one. If you count how many capital R, capital Y, small y are there, you will find only 2. If you search how many capital R, small r, capital Y are there, you will find 2. So likewise, and then if you search how many capital R, capital Y, small y are there, you will find 4. You can search the genotypes and you will see that you will come to this magic number of 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. That would give us the genotypic ratio of this dihybrid cross, which has a phenotypic ratio of 9 is to 3 is to 3 is to 1. That tells us that the genes or the characters that Mendel called as traits have their own nature. And from all his experiments, we have come to understand that they remain together. When they remain together, one tries to dominate the other. If they remain together in heterozygous state, they don't lose their individuality. And again, when they get a chance, they segregate amongst themselves and breed in new characters. So this is what is the understanding from the various crosses that Mendel did and subsequent test and deciphering of the data into what is the principles of genetics today we have. Now, as I said, what could be the conclusions? Well, we can take 4 important conclusions we can draw from this. Well, you can see the inheritance of each trait, gene, okay, is determined by units. Now, these are symbolic or factors, now called genes. Now, A for alleles, okay, that are passed on to descendants unchanged. Well, here the unchanged word is classical because we know that genes do change or mutate, but that will come later in our subsequent videos. I'll discuss with you the changeability of the gene. Okay, but as of now, the characters that are descendant depends on factors. The factors are de facto genes. An individual inherits one such unit from each parent for each trait. What does it mean? Say the trait is length, and an individual inherits one such unit from each parent. So an individual will inherit 2 such unit from his, one from father, one from mother. So the trait for length from father and the trait for length from mother. So this person will have a trait or a character called length, and for length there will be 2 genes, and these 2 genes, one from the male side and one from the mother side, that is paternal and maternal, will interact among themselves and will result in what you are. It may so happen that if your parents are tall, you might become short, or if your one parent is short and the other is tall, you might be tall, but your children again might be short, because that short gene is still within you. The third point is, a trait may not show up, as I just said, in an individual, but can still be passed on to the next generation. It might not have expressed in you, it might have remained recessive because of your dominant gene that have subdued it from being expressive, but in the subsequent generation it will show up and you will wonder where from it has come, because you might have forgotten the past generations. And lastly, the genes for each trait segregate themselves during gamete production. This is very important. Though they remain together, they don't struck friendship. They live together, they remain together, but whenever there is a chance, they live and separate out and assort themselves independently, so that new characters, new combinations, new features and variations can be created. Okay, now from all of this we understand that, and I hope that you have understood, that classical genetics is about understanding the trait, now called genes, and I've already discussed with you the gene, where of it, what of it, and how of it. Now the genes remain in pairs or alleles, The alleles behave in homozygous or heterozygous ways. They behave in dominant or recessive ways, or in codominance or incomplete dominant ways. And of course, they behave in self segregation and independent ways, independent assortment ways. So we can conclude that all the characters that we see are but fluid in nature and are constantly changing, and because of that change there is variation. And these experiments have given us these fundamental three laws. The first law, law of dominance; the second law, law of segregation; and the third law,